Good afternoon, everyone, and welcome to Oregon Sea Grant's Careers in Science Investigations webinar. This is the sixth of a series of webinars that we've been holding since April to connect students like yourselves interested in marine science with professionals working in the field. My name is Lindsay Carroll, and I am the Marine Education Coordinator with Oregon Sea Grant, and I traditionally coordinate hands-on, live, in-person programs out of Hatfield Marine Science Center. Hatfield is unfortunately closed right now, which is why we are very excited to offer you this program virtually. We encourage you to ask questions of our presenters throughout today's webinar using the question and answer function located at the bottom of your screen. It is labeled Q&A. So if you have a question, feel free to type it into that question and answer box, and we will get to as many questions as time permits after each of our presenters' presentations. Before we get started, we want to learn a little bit more about you. So for those of you that have joined us previously, you're quite familiar with our two poll questions. So Kate Goodwin, our Special Projects Coordinator, is going to launch our first poll question that's going to ask you to indicate which grade level you are in. So go ahead and take a few seconds and let us know if you're in elementary school, middle school, high school, college, or are you adult?
I'm just going to give it a little more time so everyone can figure out how to answer before ending the poll.
Thanks,
2 more
Kate.
seconds.
All right, so it looks like we have 11% that are in elementary school, 37% in middle school, 37% in high school, 5% in college, and 11% are adults. Great, thank you so much. Kate's gonna go ahead and launch our second poll question, which is gonna ask you to let us know where you're joining us from. So are you from Oregon? Are you from the West Coast, not Oregon? Are you from the Central US, the East Coast, or outside the US? So take a few seconds and select your response.
Okay, I think we have those who are interested in voting have all contributed. Oh, there's still some more. Oh, okay.
Thanks, Kate. All right, so 68% are from Oregon, 9% are from the West Coast, not Oregon, 9% are from the Central US, 5% the East Coast, and we have 9% outside the US. Welcome, my friends. All right, well, thank you so much for giving us a little bit more information about you. We're going to transition to our first speaker. We have Erica Fruh, who is a fisheries biologist with NOAA, which is the National Oceanic and Atmospheric Administration. She and her team use AUVs, which are autonomous underwater vehicles. They use those on worldwide research cruises to learn more about ocean species and habitats. So we're very excited to have her here to tell you more about it. So Erica, go ahead and share your screen, start your video, and start your presentation.
Okay, so this is a little bit weird in that I'm a fisheries biologist, but I'm going to be talking about technologies today in fisheries research. And when I started out as a biologist, I did not know that I would be the one learning how to rewire things or learning how to computer program or that sort of thing. So my job has really changed from the day I started to where I am now. But I work for NOAA Fisheries, as Lindsay said, with the Northwest Fisheries Science Center here in Newport, Oregon. And I'm going to talk a little bit about some of the technologies that we use. So you say, okay, how do I apply technologies in fisheries research? Well, if you like video games, I have a job for you as an ROV pilot. If you like movies or photography, I have a job for you in fisheries science. working with underwater cameras or videos to make shows like Blue Planet, or to support these telepresence webcasts that some of our ROV ships are able to do with live stream videos to YouTube or to Facebook. If you like computer programming, I have a job for you there also as a fisheries software developer. This is one of our softwares that we had built for us specifically to help analyze images that we take with the AUV and help us to count fish and corals and just keep track of what's in the image. And if you like robotics, well, you could be an AUV designer, an AUV pilot, or an AUV engineer. So these are sort of submarines, but they're technically robots because we program them and let them go and wait for them to come back with all the data they've collected. So in fisheries biology, we've come a long way from pencils and clipboards. When I started with NOAA, I was a biologist on the trawl survey. So this is on the back deck of a commercial fishing boat. And as you can see, Stacy's using a touchscreen ruggedized laptop computer. She's got an electronic fish measuring board. We have a barcode scanner to quantify all of our samples. And all of this talks to each other through a Wi-Fi network that we've set up on the back deck of a commercial fishing boat. So that's pretty techy, if you ask me. So the idea is when we fish on the trawl survey, we get a table full of fish, and the idea is to turn that table full of fish into data as quickly as possible and efficiently. And all of these various technologies and a little bit of Wi-Fi applied, and that's what we're able to do pretty quickly. Some of the other technology that we use in the groundfish trawl survey is we put sensors on every net that goes in the water, and these sensors report back to the ship and tell us how the net is fishing, what the temperature is down on the bottom of the ocean, what the light is, the intensity of light down there, how many fish are going into the net, all sorts of different information, and it's all sent right back to the ship, and you get a display in the wheelhouse of the boat to show you what's going on with your net at the bottom of the ocean. My current project, I did the trawl survey for 10 years and then I switched over to AUVs, and this family tree here is of some Woods Hole vehicles, and on the one side we have ROVs. And the difference between an AUV and an ROV is ROVs are attached to the ship via usually a fiber optic cable, and they are driven from the ship. You have actual pilots and real-time video. So they're really handy for making sample collections and adjusting on the fly. The AUVs, however, are unattached to the ship, and they are pre-programmed and sent in to go do a certain track, and then they can stay out for a couple of hours or up to several months. Some of the water column gliders can stay out for months at a time. They just come back to the surface and transmit data and go back on their routine. So the seabed AUV is specifically the one that I work with. And it's right in the middle of this bunch of AUVs. So here's ours. It's a twin hull hover capable AUV that can go down to 2,000 meters. It's unique in that we set an altitude and it's able to track over the bottom and maintain that altitude and adjust as it goes up and down over boulders or down slopes or things like that. So it's also nice in that we can change the sensor payload at any time. So put different cameras, multi-beams, oxygen sensors, all sorts of different things on it, whatever we can hang on it to collect more oceanographic information. Ours currently is set up with 2 downward stereo cameras, and so they point directly down. And then we have an angled camera in the middle that kind of gives us more of a side view of fish. And the graph here shows how it tracks over the bottom, with the blue line being the AUV and the pink line being the actual ocean floor. And it just kind of follows every blip and bump in the floor and maintains the altitude you set. And we take a photo every 10 seconds or so, 8 to 10 seconds, to cover pretty big tracts of the ocean floor. We've launched the AUV from all sorts of vessels. We've been able to do it from 40-foot ships up to 225-foot boats. Our specific AUV has a 6 to 8 hour endurance, and it runs on 16 laptop batteries. We can communicate with the AUV while it's on the surface, and we can also track it when it's on the ocean floor. But we don't get the data until we bring it back onto the ship and are able to download it. Here's just a little diagram of the sensors and everything that's located in it. As you can see there's a lot of foam in the upper pontoon and the brain, and then the heavy stuff, batteries, are down in the bottom. That's to keep it upright. Here are some images that we've taken with the AUV, and it can be pretty challenging to identify some of these fish species just from a top view. The flatfish, I like those. Those are pretty simple. We also, from the images, we use those for habitat classification. As you can see in the one image, we have boulders and cobble, and the other we have mud. So there's lots of different habitats down there. So we're looking to see what species can be found in which of these different habitats. We also find other stuff that we weren't necessarily expecting. We come across marine debris occasionally. This is also a picture of a sea lion skeleton down in the bottom corner there. And I'm still waiting to find my pirate treasure chest. That's my future retirement plan. We've been able to travel with the AUV. We co-own it with Pacific Islands. So we've had the opportunity to take it all over the South Pacific. The bulk of our cruises have been up and down the West Coast here, but we pack it up into 8 boxes and can ship it all over the place. So this is just one example. This was a cruise we did off of Northern California looking for corals. And these are some of our pictures. The top ones are black corals, and the bottom one is a bamboo coral. And some of these deep sea corals have been aged up to 4,000 years. Here's a sponge reef we did off the coast of Washington. And these are big habitat building sponges. And as you can see, lots of fish really enjoy using that as some protective cover. And these sponges have grown over time to almost build a sort of a almost reef like corals do in shallower waters. We've been down to Channel Islands National Marine Sanctuary. This one specific cruise was looking for different corals down there. We've been able to go with Department of Fisheries and Oceans in Canada and use their ships, and we were able to take it out to Cobb Seamount, which is 200 miles off the coast of Oregon, just outside of the US EEZ. And there's a seamount there that comes pretty close to the surface, and we got to see some really different stuff out there. We've been able to go to the South Pacific. Like I said, this is American Samoa. We did some fisheries research out there. I got my first giant clam, as you can see in the photo. That was really cool to find in an AUV picture. We've been to the Northern Mariana Islands, and you can see a crown of thorns sea star there, and my first Nemo fish in an anemone. So that was pretty exciting, because at the end of the day, I'm still a fish geek, even with all this technology. We've worked in Hawaii, in the Hawaiian Islands, doing fisheries research. we were able to put video cameras on the front of the AUV in this instance. And so again, a different sensor, a different payload, and get a different view of the world. We've taken it to the Gulf of Mexico. This was for an untrawlable habitat study. And we found some lionfish where there shouldn't be lionfish. Here is an image of a typical track for the AUV. And just we do zigzags, we do straight lines, we do grids. So you pre-program it before you send it down. This one is a track for a photo mosaic. So these lines are super tight with lots of overlap. And you take a lot of images. And then once you do that, you're able to turn it into a poster size image. This is a photo mosaic of an airplane wreck that's on the seafloor in the Channel Islands Marine Sanctuary. So this is a whole bunch of images stitched together to make a larger image. Another technology that we're looking at is automated image analysis. If we can have a computer tell us, even just sort out which images have fish and which don't, that would make a faster data turnaround for stock assessments. Some of the really cool technology that I get to work with are these big working class ROVs that can go down to 4,000 meters. They do these great telepresence cruises. You could check out nautiluslive.org or Okeanos Explorer. They all present live stream. These ROVs can dive for days at a time. Here we are launching some of the Hercules and Argus, the ROVs, on the Nautilus. The ROVs have great sampling capabilities. They can bring back corals and invertebrates. We have, my favorite is the Slurp. It's basically an underwater vacuum cleaner. Here are some of the great images. They have super duper HD cameras on them that take incredible photos. It seems like there's always a question about how many boats and which boats you've been out on. So here's the top of my head list about some of the boats that I've been on. And there's always a question about if you get seasick, and the answer is yes. It usually goes away in a day or two. And how I got here, I did my undergraduate at Auburn University. I worked for Maryland Department of the Environment for a little while after that before going to grad school at Oregon State and getting an MRM, Marine Resource Management degree. I worked as a fisheries observer for Pacific States, and that turned into a job with Oregon Department of Fish and Wildlife, and that led to the trawl survey with NOAA, and I've been there for the last 18 years. My suggestion is, if you want to work in this field, is to intern, volunteer, and talk to people who do what you think you want to do. Find out what they do on a day-to-day basis, and just try to apply what you want to know and see if that's actually what they're doing. NOAA has some undergraduate intern possibilities and NFS REU program. And if you really want to learn about AUVs more, there's a cool AUV Antarctic under ice mapping video on YouTube if you want to look that up and have some spare time. So I think that's it. If there are any questions.
Thank you, Erica. It's great to get the insights to what it's like to work with underwater technologies. So thank you so much for your presentation. We actually have quite a few questions. One of them, there's a couple of parts, but I'll break it down for you so it's a little easier for you to answer. We have a question from Sydney. She's curious on how long, like the average build time it takes to build an AUV or build an ROV. I'm sure that differs.
Oh,
how much it costs.
The ROVs are much, much more intricate and much more sensitive and much more expensive. I've heard of some of them costing upwards of $5 million. Our AUV is a little bit cheaper and a lot less sophisticated than some of the ROVs. Ours you could probably get for about $500,000. Build times depends. I've heard of ROVs taking years for construction and testing and just trying to work all the kinks out. An AUV like ours, you could probably get within a year.
Very cool. Thank you for that insight. The second part is about like the differences in their designs. Are they designed to accomplish different tasks or jobs?
They are designed to do different jobs. The ROVs have the bonus of being able to collect samples, whereas our AUV can only take images. So we just get pictures, and sometimes that can be a challenge when you have a photo of something, but you don't have a sample to compare it to have actual genetics or even the fish in your hand to key it out more thoroughly.
Great, thank you. We have a question from Lauren, and I think this is referencing the fish maybe photographed in your earlier slides, or one of your earlier slides, about do you have a specific type of fish that you catch? So maybe referencing one of your first slides.
Okay, well, I worked on the groundfish trawl survey, so we did basically everything that's not a salmon on the West Coast. All the commercial fisheries out here, we call them groundfish. It's mostly rockfish, any of the flatfish. The hake survey or hake fishery is a large one here on the West Coast. So I always say I work on anything not a salmon. And my favorite type of sea animal, I see that as one of the questions, I would have to say all the fun deep sea guys with glowy eyes and big gnarly teeth.
That's awesome. It must be pretty cool to get images from the deep sea because that is part of what you focus on too, correct?
We are down to 2,000 meters, so it's pretty dark down there and lots of cool critters.
Very cool. And we have a common question for a lot of our webinar presenters. What's the longest you've been at sea?
The longest I've been at sea has probably been 20 some days. Usually the ships we use need to come in to buy more groceries. And then you get a break. So while they're buying groceries, you get a couple days off and then you go right back out. So I've done cruises of up to 60 days, but you do get a couple days break during that time period.
Great. And we're going to squeeze in one more. Where is the furthest you've gone from Oregon?
Probably Guam or American Samoa were pretty big trips. So South Pacific.
Very cool. Thank you, Erica. And thanks for your fantastic presentation. If you had a question and we didn't get to it, we're going to work with Erica after the fact and have her answer all of the questions we haven't gotten to. So if you have any follow-ups, feel free to type those questions into the Q&A as you think of them. We're going to transition to our next presenter. Our next presenter is Alexa Kownacki, and she's a graduate student in Fisheries and Wildlife at Oregon State University. And she's going to tell you about her experiences that led to graduate school and how through her current research, she is trying to understand population health and habitat use patterns of common bottlenose dolphins off of California. So Alexa, go ahead and share your screen, your video, and begin your presentation. Thanks.
Hi, everyone. It's awesome to be here today. Thank you for inviting me. So rather than just talking about my research, I wanted to give you a little bit of a welcome to research and what research can look like in the hopes that some of this might connect to you. So I want to give you a little bit about me first because I am a human being, not just a PhD student. I grew up sailboat racing. That was my #1 passion. And I loved all things water. So people should have seen this coming, but I didn't. Today you'll find me traveling with my camera, cuddling with my dogs, and also conducting my research. And there are a few reasons why I point this out, not just that I'm a human, but I point out that I have hobbies, primarily because your hobbies are also skills. So these skills will help you down the road no matter what they are. I like to give the example that my first internship was collecting through mist netting, which is like a kind of like a tennis net where you catch bats. And I got that internship because a graduate student saw how I tied knots correctly. So maybe like Erica said, you play video games and you're keen to do, say, flying drones, which we use in our lab. Maybe you like photography like me, so you have a leg up on shooting at the, like taking the photo at the correct place and having a high quality image. Maybe you're great at art and therefore can depict things much better than someone like me who was not at. So just want to remind you to do what you like, because there will be something that relates to that. So I throw this in because plans change. If you had asked me any age between 4 and 20 what I was going to be, I would tell you a trauma surgeon or an emergency medicine physician. I love that hands-on approach to like learning and fast-paced tasks. And I loved helping people. And I was honestly just fascinated with medicine in general. My parents didn't like this because I would watch all of those TV surgeries, trauma, life in the ER shows back in the 90s and through today, and they couldn't stand any of it. Back then, I would have never guessed where I am. Path has changed, and I think that's a really important part to embrace in your future. So right now, you may think you're going to do one thing. 10 years from now, maybe something else. 10 years later, something else. That's great. You don't have to be just one thing in your life. So I'm bringing it back. We're moving forward now. So in high school was kind of my first interaction with wildlife. I saw a need because I heard about a spider monkey sanctuary in Mexico that needed assistance caring for and understanding the behavior of their animals. So all these animals are injured and abandoned or confiscated illegal pets that the Mexican government has brought to this sanctuary, where they receive care until the animals can pass away in a safe environment, because these animals are not okay to be rehabilitated for wild releases. So I ended up founding a club in high school with the help of some awesome teachers, where we took primatologists, so people who study primates like monkeys, animal behavioral specialists, and high school students to visit the sanctuary, help gather information for husbandry, and also just learn how to take data. That was my first time ever collecting what now is my life. So there's another type of research called clinical research. That's what I was doing through college because I thought I was going to become a trauma surgeon. And it was an incredible experience. So I helped run a student-run clinic for underserved communities. It's called Shifa. In Arabic, it means health. This clinic was part of UC Davis that has student-run clinics. That's where I went to undergrad. And I got to do the things that like a medical assistant or even a nurse would be able to do today. I don't know how they let us do this, but we would take EKGs, we would take vitals, we would collect medical histories. We were working with patients and physicians and phlebotomists, who were the people who draw blood. It was truly an incredible experience. I wouldn't take it back for anything, but it was one of the aha moments that this maybe wasn't my lifetime goal. And that although I liked helping people, there might be something else to it. So then I transitioned to doing more lab work. So at the top is actually a photo of one of my lab notebooks from when I was in college. And you see that there's a lot of like some pluses and minuses and scribbles. Scribbles are important to science. And those are actually saying if it's positive or negative to Lyme disease, because I used to read and look at blood work tests from wildlife and humans. So if you were to go and have to get a test for Lyme disease, which is caused by a tick, you'd be getting the same test done that I would run on wildlife. In the other photo, you see me holding what looks like a tiny baby kind of ugly mouse. That's an Amargosa vole, which looks kind of like a mouse, that we had bred because it's an endangered species. And so we were part of an endangered species breeding program where I took care of animals, part of the husbandry aspect of lab work twice a week. Twice a day. Twice a week would have freed up my schedule a lot more. And then the bottom photo of me is most recent, which is me working at a lab bench. That's just where you see all of what I think of it that looks like boring equipment, but is very important and very precise. So I thought about lab work and I was like, this is like only kind of fun if I get to see it from start to finish. And by start, I mean fieldwork. Fieldwork is the pride and joy of, I would say, nearly any wildlife, outdoor job, person, anything. It's what I call the 5% of my job, but it's the 5% that give me the most rewarding and like fulfills my passion. It's the reason I do all the other, what I think of as less exciting or maybe less thrilling work. So it makes also the lab work more fulfilling because I get to see from start to finish what my samples did. So on the top of the screen, you see me using a crossbow, which would be the same as kind of like a junior kid's crossbow you would hunt deer with. But I have the special like apparatus at the front that has like a bumper on it, like a spacer. So it only collects a small, tiny amount, like the tip of a pencil, amount of blubber and skin from dolphins or whales. And then the other photos, you see me holding animals. So I've got a snake in one hand where I was trying to collect herps, which are all amphibians and reptiles. I'm holding a bird because I was collecting any ectoparasites, which are like dogs and cats might get, wildlife also, and that's how actually your pets probably get them. And then the last photo was me holding the bat from that first project I talked about. So it's kind of a backwards progression of my field work. Another really important topic is that research can take you places. I love traveling, and I've had so many opportunities because I've, one, taken advantage of those that I've seen, and two, because I research about researching abroad. So I just highlighted a few of the places that research has taken me. So I worked in South Africa for 2 summers, collecting research on individual rhinos and cheetahs. So I would go out and try to find them and see what their health status was, see how their cubs were doing, etc. That was my first time really discovering field work outside of the United States, and it just lit a fire under me, and I realized this was something I wanted to do for the rest of my life. Then I went to Australia where I studied reef fish and tidal changes of what fish were present, and also disturbed versus undisturbed habitats and what kind of fish were present in the disturbed versus the undisturbed. This is a photo with me snorkeling because we had transects, which are lines that you have to survey. So I would snorkel every day, twice a day, and I swam next to plenty of sharks that are just nice, docile, friendly creatures. Then I went to Tanzania where I studied human-wildlife conflict because elephants there would trample into the farms of some of the local people and the local tribes. And then people in the tribes would want to hurt the wildlife because it was killing their property, how they were feeding themselves. So we discovered a way to prevent elephants from raiding the crops, which is using beehive fences, because elephants are naturally scared of bees. And these fences also then had the hives that were harvestable, that made harvestable honey, so the farmers could also then use the honey and sell it. So it was a really nice way to see how humans can work with wildlife and both can be somewhat happy together. And lastly, I went to the Antarctic to study zooplankton. So I didn't always study the big charismatic megafauna. I got to study the little things, and it was amazing. I worked weird shifts. I worked from midnight to 12 p.m. every day for 8 weeks, and I loved it. I think I was the only person who didn't get seasick, so I guess all my sailing experience helped. So my career progression a little bit after college, I did it by species, but that's just because how my jobs worked. Like Erica said, volunteering really is your way into any field, especially doing marine research. So I first started as an intern at NOAA Fisheries down in San Diego, where I counted penguins on maps, kind of like actually very similar to the maps that Erica made that were all stitched together. And I'd look for these little dots that were penguin nests, and I would click on them. So I counted penguin nests from a computer. Then I was able to go and work collecting zooplankton in the Antarctic as a paid position. So that was a leg up after 6 months of volunteer work. And then I got a position studying whales, the gray whale migration, which if you live in Oregon or the West Coast of the US, hopefully you've heard of, because they're probably going right by the coast closest to where you live. And these were all part of my journey to get to where I am today, which is PhD research. So you can see these are the species I study. These are common bottlenose dolphins. From now, I'm just going to say dolphins because that's too much of a mouthful. Where I go out on these ribs, which are these inflatable boats, and take data. So I do biopsy data, I do photo ID data, which is in this photo up at the top right. You see that there's a dorsal fin, that's the fin sticking out. Those are unique to each person or each individual, like a fingerprint is to a person. So that helps us find which animal then I can associate with the health data that I've collected. And I'm really fortunate in that my work at NOAA helped me to bridge into my PhD research. So my PhD project, at least the first chapter of it, which is what I'm going to just briefly discuss today, is a comparative health assessment of bottlenose dolphins off the Western USA, which essentially means I'm looking at how healthy the animals are and where they are and why they're there. I'm actually looking at data from the 1980s to present. So this is also before I was born, as probably was for some of you out there, which is such an incredible opportunity to have this much data. So little reasons why I'm studying this is, as I've mentioned before, any ecosystem involves both wildlife and people. Marine systems are harder to study, obviously, because people aren't always out there, or things are below the surface. Common bottlenose dolphins have these two ecotypes that have different locations in a kind of similar general area. So I like to say the coastal versus the offshore. Think of the coastal as like a plain M&M. It's easier to find because you're looking from the coast. It's still an M&M. You're going to find them anywhere. The offshore is like a peanut M&M that's like stored in someone's like hidden drawer. So you're like, I know that they're there, but they're a little farther away. They're a little bit bigger because they've got the peanut inside of them. The offshores are also a little bigger. And then genetically they're different because one has a peanut and one doesn't, but they're both M&Ms and can be purchased at the same location. So they're really, really cool things, cool species to study. So here's just one idea of what I'm looking at. So this is an aerial image taken from a drone of bottlenose dolphins. You can see on the right side, there's a mom with her calf. And you can also see how shallow the water is, right? Like this is, I think, within 5 meters easily, because you can see the ground so clearly. On the right-hand side is what my data is producing. So you're gonna see all of these purple little dots here. Those are all different dolphin sightings. And you can't tell how many there are, but there are over 4,000 in just this area right here. These other colors are different lagoons. We are trying to see if dolphins hang out closer to those lagoons or not. You know, maybe that's the cool place to go. Maybe that's where their prey are. And all of these different green lines are different bathymetry gradients, which essentially means they're like stairs. So each one is 10 meters difference. So this is a 10-meter line, and then 20 meters, and then 30 meters. And then you see the spacing get shorter between them, means there's a shelf. So it's getting steeper quickly. So I'm looking at where are these dolphins. Then if you look, you see a lot of the purple points are all within that first or second green line, meaning they're all within 10 to 20 meters, which makes sense because this is the coastal. This is the easy M&M that we see all the time. These are the photos you see of surfers with a dolphin beside them. These are these. There's also other aspects to research. So when you're doing projects, when you're working for NOAA, working for a nonprofit, you work in academia, your one project is no longer just your one project. So I am part of a fellowship program, which helps me get through grad school, but also requires a service component. So I am a SMART scholar, terrible name, I know. And this means though that the Department of Defense has said that I have to help them every summer on a project. And when I finish, I have a job working for the Navy as a civil servant. So my job is actually with the Marine Mammal Program, which is awesome because that means I'm working with bottlenose dolphins and also sea lions at one of the naval bases in San Diego. Just a quick down low on what these photos are. So the top left is actually one of my friends training one of the dolphins just on kind of floating docks. The next photo to the right are dolphins being deployed. So they're in tanks with their trainers and veterinarians on a C-17 aircraft. And then the bottom photo is another two of my friends training animals for open ocean, because these animals mine hunt and also collect items that have fallen from Navy ships. I like to point out the importance of conferences and outreach. As similar to this program, it's incredibly important to share your science and communicate it. This is why your parents, your family, your friends ask you what you're doing at school, and you should tell them, and you should talk about it, and you should have opinions, and you should have questions, and things that you think are right, maybe are wrong, say things you think are wrong. I mean, it's just important to have these discussions because this makes good science. All of us in these photos have all made mistakes, have all had failures, have all had successes. So the top two photos are my lab, and then the right photo is me presenting research in Barcelona, Spain, this past December, which was incredible. There's a few hundred people in the audience for each talk. The bottom photo is my lab, bottom right is my lab at University of Washington giving presentations to other students. And then the left is my lab mate night at Marine Science Day, which if you're a local to Oregon Coast, you know of, and has been canceled this year, but we'll be back next year. And it's an awesome opportunity to get hands-on with students and people doing awesome and amazing work. I like to touch on the importance of thank yous. So in science, but in almost any study, it's so important to thank your friends, your colleagues, your partners, nonprofits you worked for, government funding, because this is, they say, sometimes the most important slide in terms of recognizing people. So with that, I want to ask, where do you think research will take you? What are you interested in? Remember, there is no wrong nor right way to your journey. I like to give the example of I took an extra year in undergrad. I'm taking an extra year in graduate school. I didn't think I was ever going to be applying to graduate school. I applied to graduate school 3 times, and only on that last time did I finally get the funding to be here. I had someone telling me not to give up, and he reminded me of that. And I wrote my graduate school applications in a hospital because I was so sick, but was so determined. So again, journeys may take you different ways. I've had to change my plans. You'll probably have to change yours, but I want you to know that it's okay and you can have any different career path. So finally, here's a cute photo of my little splashy friends. Here's my contact information as well. I'm happy to reach out. And I ask and open up for questions from you all.
Thank you, Alexa, for that extremely inspiring presentation, for all of your words of wisdom. We do appreciate it. So I'll start off with an easy question. What's your favorite dolphin?
Ooh, are we talking like species, or like do I have a favorite of the individuals that I study?
I would go species. I think that's what they're referring
okay.
to. But feel free to also answer individuals if you'd like.
Okay. Well, some people know them by name. I truly love the species that I study. It wasn't like as a child I thought I was going to study common bottlenose dolphins, but they're really fun because you can see them from shore. I think being able to see the species and talk to the surfers and talk to the fishermen about them, it makes them like everybody knows them, and it's like a family of people who know the dolphins. So I really like my bottlenose dolphins. Very good.
We have someone who's curious, Eli. He wants to know if you studied orcas at any point in your career.
So Eli, I did in a way study orcas. So I did a lot of ship surveys. I do them still in graduate school, where we go on a NOAA ship for a few weeks, and I look for any marine mammal or seabird out there. them. And I've been lucky enough to see orcas quite a few times and help take information of them. So I did not, it was not species specific in the sense that I didn't go out just looking for orcas, but that's how it frequently happens with our transient orcas out here, is kind of they come to you because where the food is. So yes and no.
Very good. Switching gears slightly to earlier in your career path. So when you were studying zooplankton, what were you specifically looking at?
So researchers at NOAA wanted to know where the different species of krill were in the Antarctic, because krill is actually a species that's like hunted, collected. You can get like krill tablets for health related things like krill oil. So they want to know which species were where, and rather than just like dumping out all the rest of the zooplankton, we had to sample them. This means that even though we were looking at where zooplankton were, or where specific krill were, sometimes no krill, no zooplankton, but just a giant, like the size of a car jellyfish to come up, and then you had to scoop goo out. So we were looking for little tiny things, and sometimes we got really big things.
Very cool. And I'm going to squeeze in one last question, if we don't mind, friends. I know it's 4:46. Can you identify specific dolphins by their sounds?
Actually, yes, you can. My friends just put out a paper from the Navy about this. You have to know which dolphin it is making which sound to get a baseline. But not only do dolphins have their own individual sounds, family groups will, and groups in just like slightly different places. So technically, yes, I think we're getting there after this paper came out in terms of wild animal perspectives. But in captive animals, yes, absolutely, they have individual noises. So it would make sense that yes. Really good question.
Awesome. Thank you, Alexa, for your fantastic presentation. I know there's probably not enough time to answer all of our questions about our lovely marine mammals, but if you have any additional questions, go ahead and put them into the question and answer, and we will work with Alexa to, I'm trying to multitask here, excuse me. We'll work with Alexa to answer those questions, and we will post the answers to the questions and the webinar recording on our website, the Career Day website. So feel free to check back and check for those postings. Before you go, we would love for you to give us your feedback. We've been trying to use it to inform future presenters. And so if you want to go ahead and complete this survey, Kate's going to go ahead and drop that link into the chat box, which is separate from the question and answer. You can also wait when we end this meeting. There will be an auto prompt that will prompt you to fill out the survey then. We also encourage you to register for our future webinars. We have webinars every Wednesday at 4 p.m. through June 10th. And so next week, using that survey feedback, we've had a couple of folks interested in hearing from people working in ocean conservation, plastics in the ocean, marine debris. And so we're going to be hosting 2 individuals working in the field. We'll be hosting Dorothy Horn, who is a graduate student at Portland State University, who's researching the effects of plastics in the ocean and on the organisms that live close to the seashore. And we'll also be hearing from Brianna Goodwin, who works with Surfrider Foundation, which is a nonprofit organization. We haven't hosted anyone from a nonprofit yet. And so Surfrider is actively working to protect the ocean, beaches, and waves so that we can all enjoy them. So she's gonna give perspective of what it's like to work for Surfrider, a nonprofit, and all of her work focused on marine debris. I also wanna give a quick shout out to the June 10th presenters. We have Dr. Tim Miller-Morgan. He works with Oregon Sea Grant, and he is our aquatic animal veterinarian. So he's the one that ensures the health and welfare of all of the aquarium species at Hatfield Marine Science Center and also at other science centers along the Oregon coast. So if you're very interested in working with animals in your future and potentially pursuing a career in veterinary medicine and maybe specializing in aquatic animal health, we encourage you to join in 2 weeks. We also will be hearing from Michael Moses, who works for the Oregon Oregon Department of Land Conservation and Development. That's a state agency, but he's gonna he's going to cover some of his work leading up to his current position, which focused on ocean acidification. So we had a few responses requesting folks that worked in ocean acidification. So if that was you, be sure to join us in 2 weeks. So you can register and learn more about all of our upcoming webinars on our Career Day website. So if Kate wants to drop that link into our chat box, or for those of you that have already navigated there, you know where to go. But this concludes today's Careers in Science webinar. I want to thank our presenters for their time and their insights. I hope that you are learning that it's not a straight path, that all of the different experiences will contribute to your career, and those are the experiences that help expose you to things that you may be interested in or not interested in. So thank you to our presenters, and then thank you participants for joining us. And so we hope to see you at a future webinar. So thank you for joining and have a great evening.